From Microbe TV, this is TWiEVO, This Week in Evolution, episode 106, live streamed on the 25th of September, 2024. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent, great to see you. Happy 106th episode of TWiEVO.
106, good number.
Yeah, I'm glad to be here. I'm joining you actually today, not from LD Lab Studios, but LD Mountain Studios from
I guess there's a difference, right?
from home. There's a difference. It's just a few miles, less than 10 miles. But yeah, as you know, Vincent, I love to study infection biology. I'm less excited about practicing it, but I'm joining today with a third party, an infectious microbe called SARS-CoV-2. I'm COVID positive at the moment. And
so hopefully you have a nice face mask on your microphone there to protect us all.
That's right. I don't think there's a mode of transmission yet through the electrons or whatever, through the pipeways of the internet. But yeah, a little under the weather, recovering nicely, just cold symptoms, mild flu symptoms, I would say. But social distancing, to say the least. But in a good location. So one of the really fun things, about being in Salt Lake is the divide between wilderness and work is pretty seamless. And so I'm less than 10 miles away, but really in the mountains. I don't know if you can see it in our show notes, Vincent, but I put in a snapshot of the landscape behind me. Yeah, that's current events here.
It's beautiful.
So fall has arrived, both on the calendar and in spirit here in the mountains, as the trees start to turn all these various colors.
It's beautiful. You look out your window and you see that. That's great.
That's right behind me. Yeah.
So you could take a hike, right?
I could take a hike. I'm planning to right after the podcast to sort of reflect on the conversations ahead.
And
Speaking
you
of,
won't encounter anyone, so it shouldn't be a problem.
Yeah, maybe a deer or even a moose, but
well, don't give those deers COVID.
Correct. That is possible. Cross-species transmission has been detected.
Yep.
But great to be here. How are you doing, Vincent?
I'm okay. I'm looking forward to our little live stream here, our monthly live stream. I want to welcome everybody and our moderators today. We have Andrew from New Zealand, Barb Mack from the UK, and did I see Tom? Yep, Tom is here as well, and Les joining us today. Thanks for moderating. Fantastic. And tell us where you're from, folks. We'd like to hear where you are from. We'll highlight that as we go along here.
Has fall reached you out on the East Coast?
It's interesting. Last week, this past weekend, it was nice and sunny and, high 70s. And then suddenly Monday, boom, fall is here.
Yeah, exactly. Yeah.
This is this abrupt transition. So, yeah, the leaves are flurrying all over the place and the temperatures are dropping. I don't really look forward to the freezing cold, but that's the Northeast. Well,
yeah, and the Mountain West as well.
You too,
winter
right?
on the way.
You get cold temperatures, yeah.
Yep.
All right, let's see where people are from. Mark here. Let's see if I have to adjust these things. Maybe I ought to move the nameplates to the top.
Oh, sure. Well, look at that.
So we can put the comments on the bottom.
Good idea.
Mark is in Helsinki, where it is cloudy, threatening to rain, 15C.
Ooh.
Welcome.
Welcome. Speaking of winters ahead, getting closer to the darker season.
Philip is in the Wales, UK. Good to see you.
Well, welcome.
Always raining in Wales.
Yeah.
Who else we got here? Mostly a chatter amongst the moderators. FSM is the dog from Simus, Wales, I believe, right?
Yep. Welcome back.
I remember from last time I was corrected.
Hmm.
Yes, Marge says, congratulations on 106.
Thank you. Great to be here.
We had a cool 100 at the Science Mill in Texas. That was a lot of fun.
That was fun. Rich Condit joined us. We had, um, yeah, a great time in person, in vivo.
Abdur's in Brooklyn, not too far from
Correct.
here, 19 Celsius.
Mhm.
And Philip says, get well soon, Nels.
Yeah, thank you. It's going in the right direction. So I've been taking, of course, those antigen tests, and the first one, which was 6 days ago, really lit up within. They say wait 15 minutes. Within 5 seconds, this was sort of a clear result. But now, just the faintest of bands on the COVID test.
Tona is from Long Island. Welcome.
Welcome.
Elizabeth is from West Virginia. Cheryl is from Santa Rosa, California. Pete is in London.
Welcome, Pete.
Claire is in the UK as well. Zombre is in the east coast of Pennsylvania.
Great.
Theodore is in Athens.
Welcome,
Well,
welcome.
welcome, everyone. There is a question here I want to answer
Yeah.
because it's actually relevant to Nels. FSM, the dog, says, we were
Hmm.
promised that COVID would eventually turn into a
Hmm.
mild disease. Is that happening? Well, first of all,
Ooh.
we were not promised. It was just a theory, right? There are no
Yeah.
promises in science. The idea was that it would perhaps eventually morph into a mild disease but we're not there yet because there are a lot of older people who are being infected and they didn't get infected at a young age. So here's the theory.
Hmm.
SARS-CoV-2 pops in the world, infects lots of people, including people over 65, okay? But they don't make good T cell immunity
Hmm.
because as you age, you immunosenesce. And so
Yeah.
then when they're infected again, those T cells aren't there to protect them against severe disease. Now, younger people have no problem making T cell immunity. So the older
Mm-hmm.
people have a problem and they die. And so until everyone is infected at a young age, there will still be some severe disease. And we're not there yet. I think it's going to take a couple of generations, right?
Hmm.
Does
Yeah.
it make sense now? Does it
It does, I guess. Well, and the context is important. I guess the evolutionary side of the coin would first agree with you, Vincent. No promises in evolution either. Lots of lots of lots of rolls of the dice, so to speak, and then sifting through all of the outcomes. Um, but so mild, for who? So ages, immune memory, whether you're supercharged with, I was wishing now, obviously I would have gotten that booster in a couple weeks earlier,
Mhm.
missed it, and then ended up in this more, sort of, uncontrolled immune response versus a sort of organized and planned one, in dealing with some of those symptoms. But it's, well, what a sea change from, I was just thinking about, so this is my second bout of COVID-19. The first one was after the vaccines were out, but with a kiddo, who had a very mild case, but spread it from her daycare to her parents. Um, that
Hmm.
was like a whole different, even just how we thought about it. I mean, so the vaccines really changed, brought us out of hiding in a sense, um, but yeah, so already, this feels a lot more mild to me, both sort of personally and just on a sort of population level from observations I'm making. But yeah, who knows what's next? Some ups and downs. That's the sort of evolutionary part of this as well,
So
as this
you
thing
you
continues.
you did not take Paxlovid, I presume, right? There was no need to.
No, I didn't. Yeah, and so I probably, hindsight's 2020. By the time I was kind of in the middle of it, I shouldn't have been, but I was a little bit surprised that it was COVID versus, a
Mm.
regular cold, but even though those lines might be not yet, but are starting to blur, when we think about the other coronaviruses that cause common colds. At what point, and maybe this gets to the question, do we put this in the category of OC43, all of these other strains of coronaviruses that circulate in humans and have for decades? And
Yeah,
so,
that's a good
yeah.
point. There are five common cold coronas, and I think the idea is that SARS-CoV-2 will be one of those. or maybe it will be the fifth. There are four.
Mm.
Um, but it takes time. Evolution takes time. You have to go down that dark alley and not, get in trouble.
That's right. Hopefully not too much trouble. That's the open question here. I think that we're all sort of continuing to see what happens.
But
Yeah.
you are not in the age that would require Paxlovid, so you did the right thing.
Yeah, and honestly, feeling pretty good. A little bit rough around the edges, but overall, really grateful for good health, and fingers crossed going forward.
This is your second, you said, right?
Yeah, second round. I'd say this one has been slightly more mild
Mm.
than the first. The first one was let's see, 2 years ago, a little over 2 years ago in 2022. So 2 years after the pandemic. I've been vaccinated multiple times, obviously, but I did miss. So that's my maybe take home for today is if you're in that category where I was last week, which was like, oh yeah, I'll just get that booster next weekend or whatever. Do it if you instead of say it, which is where I kind of got caught here in the timing of this. Vaccines are great. Speaking of infection biology, Vincent, and the evolution of it, that's turns out to be the topic of today's podcast.
Indeed.
TWiEVO 106. Should we jump into our paper and see where it takes us?
Yeah, you bet. In fact, I can show it to everyone. Look.
Fantastic. So this is conservation of antiviral systems across domains of life reveals immune genes in humans. And so domains of life, we're talking about all the way out to bacteria, and how there are some an emerging field, actually, I would say, of surprising links between immune defenses that we see encoded by our own cells and also ones encoded by bacteria. And so before we get into get our hands dirty here, let's talk about who did the work that we're discussing. This is from the current issue of Cell Host Microbe. Vincent, how did this paper get onto your radar screen? Why did you decide to bring it forward?
Good question.
Stumper.
Don't know.
That's okay. I've
Someone
been
may have, someone may have sent
suggested
it in.
it. Yeah.
I don't remember. But it seemed appropriate because one of the you personally have been involved in research that looks at new elements coming from, in your case, viruses, right? Retroviruses being taken over for immune regulation and so forth. So this kind of
Mm-hmm.
fits with that. I thought it might be interesting.
Yeah, no, I love it. It's one of two papers actually in kind of overlapping space in the issue. And I sort of jumped ahead here. I meant to say, so we have 4 co-first authors. I don't know if that's a record, but right up there. So Jean Curry, Matthew Howdeket, Veronica Hernandez Trejo, and Ernest Mordre. Senior authors are Enzo Poirier and Adi Bernheim. And the work was done largely in Paris, both at the Pasteur Institute and also the Curie Institute, with some collaborations, it looks like, with the Max Planck Department of Molecular Biology in Tübingen, and also the Human Evolutionary Genetics Unit, also at the Pasteur Institute. And so, yeah, I'm really glad you nominated this paper because it really fits into a growing field or a growing collection of work, which asks, where do immune defenses come from? What are the origins of them? The deep evolution, and with a specific look, in this case, at innate immunity or sometimes called intrinsic or cellular immunity. And so just to kind of step back and define a few of the terms here, and we'll also talk about adaptive immunity, which I think caught a lot of people by surprise some years ago, that bacteria sort of encode these really complex sets of different immune functions all the way from innate to adaptive. And so innate immunity or intrinsic immunity, these would be the single genes that are encoded in genomes, or in a lot of cases, gene families. Some of these families can be massive. And basically upon infection, or in some cases as part of cellular surveillance, let's say one of those SARS-CoV-2 particles fuses with a cell or a set of cells in our nasal cavity as we breathe this in and out, and starts to begin the virus replication program in that cell. Intrinsic or innate immunity starts to kick in almost immediately. So we have all of these proteins that can bind foreign nucleic acids, DNA and RNA, and sort of set off an alarm bell for the immune system. Transduces a signal or sends a signal to the nucleus of the cell with the instructions to turn on hundreds of genes, which encode all kinds of different functions that can slow down or block the virus replication program. When we think about adaptive immunity for us in our immune systems, we think about antibodies, right? So either when we get a vaccine, our immune cells see this foreign protein, this epitope, respond by basically making a protein that can bind that, and then also kick off an immune response in real time, adapting to the shapes or patterns of the viruses that it sees in real time. And I think, historically, actually, I'm really curious to hear, Vincent, sort of when you were coming up, would you have guessed that bacteria have adaptive immune systems, that they have complex immune systems?
You mean at the beginning of my
Yeah.
scientific career?
Yeah.
No, I mean, so I grew up in an era where lysogeny was a big deal, right?
Mm-hmm.
And we knew that. So in other words, a lysogen is a bacterium that has a phage genome in it,
Yep.
and
Virus
it's
genome. Yep.
silent. It's
Mm-hmm.
silent. It's a virus genome, and we knew that that would confer resistance to super infection. So there was something going on, right? But we didn't know
Mm-hmm.
the mechanisms. But I would say that molecular biology has changed that. Now we can understand mechanisms, and there's been a huge explosion of studies on anti-phage defense in bacteria
Yeah.
because the methods are amazing. In fact, just a recent TWiV, we
Mm.
we talked about one of those. This is so cool. The name of the
Mm.
TWiV is Hotel California for phage. So there is a there's a phage protein that is made to prevent other phages from infecting the same
Mm.
cell,
Mm-hmm.
and it interferes with assembly. And in particular, when the tail of a phage is put together, there's a
Mm.
protein attached to it called the tape measure protein,
Mm.
and that determines how long the tail gets. So
Yeah.
so amazing. And this anti-phage program binds to the tape measure protein and messes up the tail assembly of an incoming phage. Anyway, that's one of dozens of mechanisms, because it
Yeah.
makes sense. If a phage is sitting in a bacterium, it's not good if other viruses go in and take over, right? So there are
Yeah.
lots and lots and then of course there are bacterial defenses against phages, but none of that existed. The first ones that I remember coming up were restriction modification systems,
Uh-huh.
right?
Yeah. Yeah.
Whereas if
Yeah.
DNA wasn't methylated, it would get destroyed in the bacteria. And we took advantage of that with restriction enzymes and so forth. But today it's just amazingly
It's it,
complicated. Yeah.
exactly. It's exploded, and a lot of I think our assumptions, certainly my assumptions about bacteria being sort of relatively simple cells, single cells, not multicellular. Um, the viruses somehow seem,
You know what we used to call them? Bags of enzymes. Bags
yeah,
of
well,
enzymes.
yeah, exactly. Bags of enzymes, or the cytoplasm soup was another sort of, I think, old sort of mythology there to some degree. Um, but my goodness, no, there is so much complex biology. Probably the most famous example, antiphage example and adaptive example, is CRISPR.
Mm-hmm.
Um, right? Where these, so basically, a nucleic acid based memory of the phages, the viruses, the bacterial viruses that had infected in the past is then written in the genome of the host, so that as these things come on, there's sort of this memory, sort of echoing some of the concepts with adaptive immunity and our antibody responses, et cetera. And so, yeah, there's been tons of studies recently now, as I think, as we sort of let go of some of the old views about the sort of false simplicity um, of these bags of enzymes, these of bacteria, to see all of the cellular complexity and also the molecular complexity or the immunological complexity. And so this raises, I think from an evolutionary standpoint, raises a really interesting question. The topic of this paper is, given all this complexity, what are sort of shared immune defenses? Maybe not, the things that are happening in bacteria. Are there echoes of that or expressions of that in our own cells, in our own immune systems? And so the evolution here, there's three sort of pathways. One is that these are so ancient that there were phage infecting ancient bacteria, or the ancestors of modern bacteria, that a lot of these immune functions came online. And then as our populations diverged from each other, some of those functions were present in a common ancestor and continue on for more than 1 billion years. A pretty wild and exciting possibility. There's also cases of convergent evolution, meaning that the same similar looking genes or similar looking functions independently end up doing similar jobs. And so that's definitely something to keep on the radar screen here. We'll discuss sort of both convergence and divergence, and whether it's you can distinguish between them. In some cases, it's pretty messy when we think about these long evolutionary timescales. And then finally, there's a great trick that bacteria do prolifically, but has happened enough in our own sort of genetic past as well, which is horizontal gene transfer. Instead of a gene being passed from mother to offspring, it somehow gets horizontally transferred from one species to another, from one lineage of life to another. And some really fun examples of how viruses might act as conduits between these different species. In addition to sort of replicating themselves, they might be passing genes, almost like trading cards, between different hosts that they infect. And so anyway, that's sort of the genetics playing field, all kinds of possible sort of pathways. But the immunology or comparative immunology here is really exciting too. And so just to spend a moment on this, I would say in the last 10 years, there have been a couple of labs that have really been on the forefront of discovering a lot of these complex bacterial functions. This is Philip Cranches's lab. He's at Boston Children's and appointed at Harvard, and Rotem Sorek at the Weizmann Institute in Israel. They've been just prolific. It's sort of like a immunity gold rush, Vincent, in bacterial genetics to find all of these anti-phage systems, now up to more than 100 of them that have been proposed, using a combination of computational pipelines and tools. And we'll get at that and what the authors of this paper do along those lines. And then, really great functional assays to test whether if you take either you add some of these, gene encoded pathways into bacteria, or take them away, and then test whether the phage, the viruses of the bacteria, do worse in the presence of these things or better in the absence of them, meaning they replicate more if you mutate or take away some of these proposed phage defenses. And so it's almost like you pick up a scientific journal these days that has any topic in immunology, and you'll probably find a paper or two saying, hey, here's a new way that bacteria seem to be counteracting the viral phages. And then this has been going on for billions of years. And so then you've got sort of both, the time and the space, genetically speaking, to really see evolution in action.
I guess we should not be surprised at this. I mean, remember when the first reports came out, it was, oh my gosh, it's like exaptation of retroviral genes. People were surprised, but even more bacterial genes coming into eukaryotes. We should be surprised because nature makes good use of raw materials, right? If things work, they're used, and DNA is everywhere. So there's really no barrier to genes going from one kingdom to another. So we should now understand it's the way things happen.
So the way things happen, it's that balancing act here, I would say, of like ultra rare events. Not rare in bacteria, by the way, between bacterial species or populations. That's like sort of over the top levels, frequencies of horizontal transfer. Pretty rare or ultra rare in eukaryotes. But again, give enough evolutionary time, rare things that are consequential stick around. And so given that long sort of runway of trial and error here, I think that's where it really starts to, even where, I think maybe that's where, sort of the paradox in thinking, how could something that's so rare be so consequential? And evolution offers the time to sort of thread that needle. Yeah. And so, given the fact that these might be rare events, but that they, if that happens, if like, you gain a gene that's the difference between whether you're vulnerable or complete or resistant to a viral infection, you can imagine that that would very quickly, if it's successful, take over a population. There's strong selective pressures if there's a lot of pathogenic effects that can impact the ability to have offspring. And so the authors kind of set out there. So they, there's been a lot of work from the Cranches, Sorek, other laboratories, which have actually said, okay, well, here's a eukaryotic innate immune function. If we go look in bacteria using a number of tools, does that also exist there? And so there have been a number of examples of this. There's the one I'm sort of partial to is there's an immune defense called CGAS, which is alphabet soup for cyclic GMP AMP synthase. This is basically one of those proteins that's in our cells, sort of on patrol surveillance, will bind, in this case, double-strand DNA from viruses, and then sound the alarm bell through another protein called STING. So the CGAS STING pathway, just recognized for a Lasker Prize by James Chen at UT Southwestern, his discovery of CGAS maybe 10, 15 years ago using biochemistry. So this has been found now in bacteria, not just for immune defenses, but even for bacterial communication using these cyclic nucleotides. So that's sort of the signal, that's the alarm bell in this case. The host has this protein that binds to foreign DNA from a virus, and then it produces a modified nucleotide, a cyclic nucleotide, that then signals. And bacteria have been talking with to each other with these kind of molecules, again, for a really long time. And so seeing how these things get co-opted or used both for immunity and for other sort of communication functions, cellular communication functions, is pretty interesting here. So the authors of this paper decided to take this sort of train of thought in the opposite direction, because there have been, I think they propose 132 antiphage systems that are encoded by bacteria, a lot of which have been discovered just in the last few years. In a lot of cases, we don't know how they work, except that they do work. We don't know the mechanism, but we know that they have that function of reducing the ability of phages to propagate or to replicate. But so then the authors here want to ask, okay, if we start to sift through using evolutionary analysis these 132 pathways, do we find similar ones in our own genomes, in our own cells, all the way from bacteria to humans? And so to do this, they start out, they've actually in the past used their own bioinformatics tool that they call Defense Finder. And they've trained this pathway to try to propose or identify in bacterial systems some of the immune defenses. And it uses a number, I won't go into a lot of the details, I don't know a lot of the details, but they use some of the features that you might predict would be present in an antiphage system. So this includes some of the sort of protein domains that might be used in recognizing proteins or nucleic acids. So there's always sort of the sensing and affecting sort of at play in a lot of these. You first figure out that, there's a problem in the cytoplasm. There's something here that's replicating that as a host might not be super beneficial. And then with effective sensing, then comes affecting. You trigger a response. You either say to the cell, in essence, let's turn up hundreds of genes, or let's activate this protease to chop up the virus proteins, or let's target the nucleic acids to chop those things up. There's a lot of chopping that goes on, trying to degrade this thing physically before it can overcome a cell and replicate at the expense of the host. And so, a lot of these bacterial antiphage systems are also encoded in so-called operons. That meaning that there are genes that are functionally linked, are geographically linked on the chromosome or on in the DNA, in the genome. And so you find the genes next to each other or enriched in sort of close proximity that are involved in these functions. And so this has been used by several labs pretty successfully, various tools like this to propose some of these antiphage systems. Now they turn this around and ask, okay, well, let's look in the eukaryotic genomes and see if we see a similar thing going on here. Can we propose these? I think the answer is maybe from that analysis. So they say that they see some of the stuff that they might expect. But then they really take more of a candidate approach. They choose 3 of these so-called antiphage systems, bacterial immune defenses, and then search eukaryotic genomes, our own genomes, for whether they think there might be similar immune functions at play. So we have to do a little bit of, we have to walk through a little bit of mythology here, Vincent, to set up the 3 test cases of immune functions, bacterial immune functions. The first one is called Mokash. This is the name was taken from the goddess protector of women's destiny in Slavic mythology. So this is kind of one of the fun, that's a lot. So this is one of the fun sort of moments of discovery, immune discovery, is if you discover this thing and sort of show or prove to people, prove to your peers that this looks like an antiphage system, but it hasn't been described before, then you can name it, whatever you want. So there have been some pretty wild naming schemes out there, a lot of them based on mythology. So we have Mokash, and we'll talk about what that is and whether we think it's showing up in our own genomes. We have Ilios. This is the Greek goddess of mercy, another antiphage system discovered in bacteria recently. And Lamassu. I looked this one up on on Wikipedia. This is a celestial being from ancient Mesopotamian religion, bearing a human head symbolizing intelligence, a bull's body symbolizing strength, and an eagle's wings symbolizing freedom. So intelligence, strength, and freedom, which is what you would like. I would take it if I could get some intelligence, strength, and freedom from COVID-19. Count me in. I wish I would have put this together a little bit quicker. In this case, practicing some of this personal virology at the moment. But yeah, so those are the 3 systems, and by systems I mean sort of immune defenses, several genes encoded by bacteria that execute a specific immune response to viral infection. And so the question then is, in these 3 cases, or their candidate cases, can they find convincing evidence that we encode similar defenses against the viruses? So we're not infected with phages. We're infected with, obviously a sort of a whole compendium of familiar clinically relevant viruses, from coronaviruses, herpes viruses, pox viruses, influenza viruses, et cetera. But the open question being whether there's sort of crossover, given very different virology. Can some of this sort of immunity sort of, really matter even for very different classes of viruses? That's another field, Vincent, that I think that's pretty exciting, kind of maybe a few steps behind the comparative immunology, is the comparative virology, drawing some of these deep evolutionary links between all of the diverse classes of phages and then the types of viruses that we see showing up infecting our cells, and whether we can draw either these sort of deep divergent links. And there is some evidence of that, genomes, not all the way out to bacteria, but among distantly related mammals, let's say, or even animals, where there's some surprising fossilized virus genomes that suggest that there's a lot deeper sort of heritage in the types of viruses that have been affecting diverse animals. All the way to the rapid evolution that we're very familiar with. I think again we have a lot of assumptions about not just the sort of false simplicity of bacteria, but also the sort of false just viruses are changing constantly. But there's also sort of these deeper links that are pretty mysterious. Anyway,
even though hosts are divergent, right? Bacteria versus eukaryotic hosts, so the viruses are going to be different. There are some properties of viruses that are invariant. For example, the capsid.
Yes.
It doesn't matter if you're a bacterial virus or a eukaryotic virus. The capsid has to have the same function. And so we see commonality in capsid proteins in bacteriophages and viruses. We're going to talk about that a little bit today, actually.
Yeah, exactly right. Really good point. And also to point out that, these things are not held completely separate. So we do have phages, but they're infecting the bacteria of the constituents of our microbiomes.
Yep.
And so, right. And so there are plenty of collisions possible here when we think about the fleas of our fleas so to speak. Okay, so anyway, let's dive into the first example here and see if it holds water. So the Mocash proteins, again, this is the goddess protector of women's destiny in Slavic mythology. These are a set of proteins. So one has a phospholipid domain, and the other is so one has a phospholipid domain, and the other is helicase, an RNA helicase, that have been shown, still working out some of the mechanisms here for the antiphage for the bacterial side, but have been shown to block phage viruses, to block phages. And so um, what the authors do is they run these proteins through their pipeline. They note that it's really hard to sort of so you have phospholipase domains are common not just in immunity factors, but we've diversified in our own genomes. We're just littered with phospholipase domains and families of genes that are using it for conducting different cell biology. And so to try to get at a little bit of a molecular link or an evolutionary link, they start to build trees. And so the phylogenetic trees. And then what they're asking is whether you see certain members of these families of genes that are associated more closely with the ones sampled from bacteria antiphage systems. And they pull out PLD6. This is one of the phospholipase family members, which here it is. So you can see that little pink square with PLD6. So that's the tree.
Yeah.
The color coding there. So the green is just the sort of confidence they think in the sort of organization of the tree. And then to the far right, you've got outgroups. So those are phospholipase domains, but that are not sort of as close relatives. And then what they like about phospholipase 6 is that sitting around those darker squares, which are Mocash proteins from bacteria. Now, immediately, Vincent, there's a little bit of a challenge here, right? Which is, okay, they've highlighted the pink one, PLD6, but all of those other green ones around it, those are other PLDs. And so just based on this level of analysis, it's not clear to me why 6 should be pink and the others shouldn't be if you're only using the phylogenetic tree. But in this case, they're not only using the phylogenetic tree, they're going to the literature. And I think one of the sort of strengths of the group that's doing this research is they're really good at sort of fishing through all of the information that's out there, sort of curating the literature and asking among the sort of dozen gene possibilities here, which one might actually be closer to an immune defense. And so the reason they nominate PLD6 that they talk about is based on a cartoon in panel C in the very left corner of the figure you're showing there. There are all those scissors. This is part of a small RNA pathway. So RNAi or RNA interference was a really interesting sort of pillar of immune defenses. Invertebrates, in particular, use RNAi pathways, small RNA pathways, to recognize viruses, chop up their nucleic acids, and block them from replicating. There's also sort of related pathways or different flavors of small RNA pathways. The one that they're highlighting here is the pi RNA pathway, not pi like PIE, like let's have a good treat after lunch, but pi, the sort of type of RNAs, in this case, that are known in our cytoplasm, in our cells, to block transposable elements from propagating. Sort of another.
I found that really interesting. I mean, these are small RNAs involved in making sure the germline isn't contaminated by these transposable elements, right?
That's right. And so they're, drawing, these are sort of marooned. One way of saying it is sort of virus-like particles are marooned in the genome, in the germline. And, some echoes of some of the lysogeny, the insertion of phages into bacterial genomes. So that's the link they draw. So there's two immune sort of links that probably haven't been put together. And they're sort of saying, hey, actually, maybe there's some, common history here, evolutionary history between these. And so
the
that's
idea, Nels, is that if these elements moved around, they would mess up the germline, right? So you have to take care of them with these small RNAs, right?
Exactly. And so PLD6 has been proposed. It's actually a protein that's involved in some mitochondrial biology, but it's also been proposed to add some of the specificity to the pi RNA pathway. And then that connects to another protein that you find the RNA helicase, right? So in order for the scissors to work, there's another protein that's recognizing the RNA and sort of progressing along it, changing its morphology, changing it from, in some cases, double-stranded to single-stranded, or just changing its topology in ways that make it more accessible or allow other enzymes to act on it. In this case, the enzyme is a protease that's represented by those scissors in that cartoon. And so here they draw sort of a second link between a protein called MOV10L1, some alphabet soup here for you, another family of helicases that sort of falls into that same category. And so of being involved in that pathway. So it's sort of, in a sense, building a biological, almost like a circumstantial case that there is some link here. These aren't just sort of random things that are shared by the genomes, but that there might be some functional connections. And then one of the fun things, and you can see this, so all of these ribbons that you see, these cartoon ribbons, are representations of the crystal structures. And so, as we've talked about on TWiEVO before, we're kind of in this moment where structural biology has undergone sort of a revolution using machine learning, artificial intelligence in more sophisticated ways to predict from just primary sequence what do structures of proteins look like when they're folded up in 3D. And that's what these red and blue and gold pictures are representing, are either crystal structures or predictions of structures. And then trying to see whether there might be sort of hidden patterns or shared patterns from E. coli all the way out to melanogaster fruit flies, all the way out to humans. And, you can squint at it and see some definite sort of shared features here. I don't want to throw a lot of cold water, but just maybe a note of caution. If we look, for example, in the lower right panel that you're showing, Vincent, the human predictions, you can see these sort of like spaghetti scenarios, especially in the blue. That's where the prediction is sort of really low confidence, kind of unlikely that protein is really folding up like spaghetti there. But so, I'd say certainly consistent evidence as you build a circumstantial case that MoCache is encoded in our own genomes, potentially by PDL6 and by MOV10L. And that's based, so now they're using sort of the computational, the circumstantial case, and then tying that to the function that the experiments that other people have done to draw that connection in the first place. So that's MoCache. And then I guess, to take it to the next level, they picked Ilios. And so to take a closer look.
Nels, they don't do any functional stuff with MoCache, which they do with the next sets, which is too bad, right?
That's right. And so they kind of use it as they say, well, sort of proof of principle, I would say, in a sense, the way they've presented it. Whether that's true or not, I think it's open question, a little podcast peer review here. And they really are leaning on some of the, the functional work. So MoCache, I should say, I don't think its mechanism is known in bacteria. The PLD6 and MOV10L or MOV10L connection for piRNA biology a little bit better there. And then the molecular links or the evolutionary links that they show are sort of, it's all kind of swirling around. I don't think it's a slam dunk necessarily in that case.
I think they say that animals that are deficient for this picoRNA pathway, of which MoCache is a part, they're sterile because they can't deal with these transposable elements. So that's kind of one of the evidences that this is involved in defenses, right?
Correct. Now, then what they kind of conclude with, which I should have mentioned, is that given those evolutionary links, what they're proposing is that this system, the MoCache system, was active in really ancient ancestors of modern bacteria, and that sort of like was the training ground in a sense for this as a anti-phage scenario. And then that it's kind of was so successful that then as ancient populations diverged, it stuck around and then has been sort of repurposed or developed through evolutionary time to take on this role with transposable elements. Really tricky to say that. So just because there's so much evolutionary time here, and evolution can kind of break the rules, right? So, were these, so all of these families have been around. Part of, and I meant to mention this, and maybe I'll return to this idea in the second two examples. But a lot of the evidence here is based on that sort of that phylogenetic scenario. And what's important is the outgroups. So there are all of these other protein domains that are the same, but don't seem as closely related. And so the concept that the authors are putting forward is that the immune defense sort of came later. And the fact that there's a eukaryotic protein at this sort of that's related to some of these relative to the outgroups, that gives that enriches for immune functions. That's where they think their bioinformatics pipeline is useful for sort of proposing what the candidates are. But yeah, whether it's divergent evolution, like they're proposing a direct link, whether it's convergent, where later on in time, a phospholipid or a phospholipase D family member like PLD6 gets recruited in. Those are open questions, I would say, which are fascinating and difficult to get at because it's just the timescales are enormous here. Okay, so next up, Ilios 2, and we'll kind of hit on some of these same themes because they're using their same pipeline here, in this case, to pick up on a different set of proteins that are also antiphage. So these are GTPase proteins, and I love this. So this is like, when I was coming up as a cell biologist, spent a lot of time worrying about GTPases. These are proteins that can really sort of the grease in the gears for cell biology, to move around membranes, to give compartments identities, for all of the membrane trafficking that our eukaryotic cells depend on. We're really, GTPases are the core of a lot of the cell biology here. There are also some really fascinating cases where GTPases have been repurposed or sort of recruited, so to speak, to as immune defenses. And so GTP is a type of nucleotide, and it's the turnover from GDP to GDP that is sort of the enzyme functions that kind of give this specificity or this identity. That's the enzymatic activity. And then you can sort of decorate these proteins with other domains that bring sort of specific functions or targeting to them. And so we've known for a while that there are these they're also called dynamin-like proteins, which is a family of GTPases, large GTPases that can be involved in blocking virus replication in our own cytoplasm. So a set of proteins, MXA and MXB. These are different ones. It's a whole different family of proteins. And they use their pipeline again to draw a link between the Ilios GTPases that you see in bacterial genomes with the so-called GMAPs, GIMAPS. These are GTPases of immunity-associated proteins. They were named that a while ago. Not a lot of study, actually, of GMAPs in animal systems or in human systems as a potential immune function, but they've been linked to some of the development of white blood cells, T cells, I believe, in some of the developmental steps. They're also noted to be upregulated by interferon. So this is one of those alarm bells when you sense that a cell is infected. When our cells do this, they turn on hundreds of genes, including some of these GMAPs. And so this is, again, one of those sort of circumstantial cases where there's been just enough of a foothold on kind of the human side of the story where it's not surprising to see the GMAPs as with a link to Ilios, given sort of the idea of a shared immune function here. This is like, I'm a big fan of GMAP, so we stumbled into them by accident in some of our own work in my lab. We were messing around with zebrafish, and we discovered, we've talked about this, Vincent, we discovered a picornavirus of zebrafish, a natural virus. And when we looked at the genes that the infected fish were turning up relative to the uninfected fish, we noticed that some of the GMAPs that the fish encode are turned up. And so we looked at that and we got really excited because humans, we encode 8, I believe, of these GMAPs. There's genes. Fish can have as many as 150 of these things. And so fish have just absolutely gone bonkers on the number of gene copies here. This also illustrates, again, this sort of, like, depending on the animals you look at, the size of the gene families involved in immunity, it's almost like a accordion. It's breathing. There can be hundreds of genes in one sort of lineage. In this case of fish, if you go to bats, there might be 10 of these, and humans 8, and it's just sort of a different ball game. And so under the hood here, there's a lot of sort of dynamics and protein family evolution involved in immunity.
So now, from your results, you think that some of these GMAPs in zebrafish are having an antiviral role, right?
Absolutely. But what it is, we don't know. And so that's why I was really kind of excited to see them draw this link between this sort of ancient anti-phage system with the GMAPs. I think this is a family that deserves more attention for possible interesting immune defenses. So they give it some attention. So they narrowed down so that you can pick 8 of these genes. They picked 2, GMAP 5 and 6. Again, how they picked those ones over the other ones, actually not clear to me. But they decided to do some virology. So they delete them, ironically, using some of the sort of tools out of the bacterial immune systems. I don't think they were using CRISPR, but they're using some silencing. That's a small RNA pathway. Using sort of immune systems for our technological, advances here in a sense. So they disrupted GMAP 5 and 6 in human cells, and then for reasons again that weren't completely clear, decided to test herpes virus 1, HSV1, to see what to see. Yeah. I was going to ask you. I'll get here in like a second. I'm curious as the virologist to get some of your impressions of the virology. But basically, they have some evidence that fewer cells get infected, and when they do, fewer viruses are produced. Pretty subtle. I think the virology here feels a little bit tacked on, a little bit slightly out of context. But what they did do, an experiment that I liked a lot, was they took one of the GTPase domains from Elios, the antiphage system, made a chimeric protein with the enzymatic domain, and then put it with the rest of GMAP6, and then asked in their assays whether that appeared to be active. And the answer was yes. The bacterial GTPase domain with the rest of the GMAP6, our own gene, spliced together and then overexpress, reduced the number of herpes virus, or the frequency of herpes virus infected cells.
Yeah, I mean, it's a modest effect, right, Nels? And you wonder if almost anything would have a similar effect. I just don't know.
Well, that's the question. So what I think you would ideally, and so, but let me step back and say, the advance here is less, I would say, in really nailing the virology. It's not close, but that the advance is that pipeline, right? And sort of, and drawing all this information together in new ways. I went to the website for Defense Finder, their tool. And in addition to sort of, kind of analyzing or proposing new anti-phage systems, they also do a nice job of sort of curating a lot of the data. So it sort of lists those 130 or so systems that are known, links it to the papers, and sort of gets, sort of a clearinghouse for information, bringing it together. But but beyond that, the virology, so there's modest effects in the amount of viruses,
right?
And then, and then also whether just the number of viruses, you start to worry about non-specific effects, especially when you're messing with the cell biology, right? The GTPases are known to mess with membrane trafficking, and sort of as a side effect, there might look like a specific immune function might just be some cells that are sort of messed up. And so
Nels, would you agree with their conclusion that these data show that GMAPs from bacteria thwart herpes infections? I mean, thwart?
That's a tough one. A little podcast peer review. No, I think that's thwart is a strong adjective or a strong verb.
Really means block, and they didn't block it here. They reduced it, but
no,
I think biological relevance isn't clear, right? That needs more work.
I think it needs more work. Why they picked herpes relative to, I think ideally you would pick a panel of viruses. Yeah. So we don't, again, we talked about this a little bit. We don't have we aren't infected directly with phage.
Yeah.
There definitely are some, as you're pointing out, some really core connections in virology, whether it's capsid, whether it's some of the polymerases that are working on the nucleic acids, those are shared. But then there's so much divergence in the viruses. And so the way they just sort of move immediately to herpes virus was not really motivated in the
Yeah,
manuscript in a
yeah.
way that kind of made sense.
I
You
mean,
know?
they do note that VSV was not affected by their
Mm-hmm.
GMAP 5 and 6 changes. Um, but I agree with you. They should have done a panel of viruses because just picking one randomly or two. I mean, I think they picked herpes because the capsid fold is shared with the phage capsid fold, right? And so
Yeah,
they
and that
figured
come
this makes
exa-
sense.
that
You
makes
know, they're,
sense. Yeah.
they're DNA viruses, but it's kind of random still.
Well, yeah, it felt a little random. That's a good way to say it. Lama, like the herpes connection comes up in Lamiso, the next one that we'll do. And then it almost makes you think, did they do that one first? And then they were like, oh, well, let's throw it on the Elios sort of,
Yeah, maybe. Yeah.
like that's another possibility here. But anyway, I think, what I would say is casting some light on GMAPs, count me in, because I think this is an underrated possible immune defense. And so
Mm.
if this motivates other labs, I'm trying to motivate my own lab to study the GMAPs in more, so with some more vigor. I think it would be really fun to see how this might sort of add to intrinsic or innate immunity, another family of GTPases that might be on the act, sort of like that circumstantial cases building. But can someone with a set of experiments or a way forward really kind of nail it and find a really robust immune response under sort of various situations? So yeah, connection between phage and herpes viruses a little bit, I think, in the ether here to say a little bit. And then again, the notion of is this were the Elios sort of ancestors shared in our common ancestors, and then they diverged, maybe. But there's also, again, all of this possibilities of convergent evolution. When I just think about the hundreds of G maps in fish alone in a sort of shorter evolutionary interval, the notion of convergent evolution very much seems possible. Or it's sort of like they look the same. It's almost like we're glimpsing at the fins of fish and the fins of whales, and it's like, well, they're both moving those critters along underwater, and so that looks really good. And then, or along the way, was there horizontal transfer? Did the Elios genes, were they donated between, in either direction at various times in various populations? And then we kind of come back and we
Mm.
see the modern species of bacteria, the diversity all around us, which is vast. We see the modern diversity of eukaryotic systems, which is not quite as vast, but not too shabby. And then sort of, who gave what when, I think is still a very interesting and open question. Okay, so then on to our last one, Lamassu. And, if you haven't already, Lamassu, do a little Google search, the images there, these sort of wild human-headed, eagle-winged horses that you might find outside like a stonemason temple, or in Mesopotamia, where the original artists sort of got to work here. And so this one is kind of fun, Vincent, because it brings up another feature, sort of a core feature of a lot of the anti-phage systems, and then sort of sees how that might overlay onto eukaryotic immune evolution. And this is the idea of microsynteny, or for bacteria, for operons, the idea that there's a link between genes that do similar jobs, or part of a complex that does a job, a pathway, and the geography of where they're located in the genome.
Mm.
And so, that allows for bacteria, that allows for co-regulation. If you have three genes that come together in a complex to do some of the bacterial cell biology, they might be turned on or regulated at the same time because they're in proximity. And a lot of the fundamental molecular biology that we were hinting at a little bit earlier in the run up to this, really like showed that in beautiful ways, the early work of how bacteria sort of have these elegant systems in place to co-regulate genes that are involved in some of the same functions. Okay, so microsynteny, that would be the idea that the geography or the location of genes, they might be more close to each other, also in these eukaryotic systems, or shared. Synteny is shared, basically the same locations of genes next to each other among different species. That would be the idea of genes being
By the way,
syntentic.
in bacteria, that's often how people discover new immune genes because they say, oh, we have one here, and then next to it, there are a bunch of genes, and they
Yeah.
turn out also to have immune functions as well.
Exactly. And so that's another way to build a kind of circumstantial case, is then you take a set of genes that have some of the homology or the phylogenetic relationship sort of more than once, and then do they sit together? And so for Lamassu, the authors think that that's the case. They find 3 genes in our own genomes, and we're about to wade into some more alphabet soup here. So get your spoons. We've got FHAD1, CTRC, and EFHD2. First one is forkhead associated phospholipoprotein binding domain 1. I think the soup is as good as the full name in this case. The second one is chemotrypsin, a pretty famous protease, one of those scissors that was in one of the cartoons previously. And then hand domain containing protein D2. A lot of alphabet soup. But what's interesting here is that so the genes are located next to each other. There's, again, a little bit of immune sort of link that they can draw from the existing literature out there. So EFHD2, the third gene that's sort of in the collection, there's evidence that it binds to HSV1 capsids. And so this is putting together, in this case, that sensing or that recognition to then an effector, which would be the protease, and then somehow FHAD1 playing in the middle, and or orchestrating this sort of between them. So they work to again express some of these genes in human cells, and then see whether that affects HSV1, herpes virus 1 infections. Ran into a little bit of some roadblocks here, so they find that they can't express all 3. I think the FHAD1 in particular was not behaving in their sort of overexpression systems. That's not unusual. And so they, do sort of kind of a hodgepodge of virology or molecular biology to look in different cells with different sort of flavors of knocking out or knocking down one or the other components, and then seeing some subtle effects. This isn't, I think, the strength of the paper, but drawing that evolutionary connection in the first place, and sort of hopefully motivating future work on the infection biology of this complex could actually go somewhere interesting.
Yeah, it's interesting. I guess they only checked herpes and VSV, right?
Yeah. And so that's, a little, again, I think there's more to the story here, and with some, with some careful virology, some panels of viruses hitting a few different classes, a few different cell lines.
I think, as you said, the effects are really modest. So it's, I don't know why, right? They do try different systems, but
correct.
1.5 to 2-fold, 50%, just not really gangbusters.
No. And there again, you worry about specificity. Are you is it just messing up the cell in some way versus, and there's some controls you can do, et cetera. I think what you would really love is sort of that, some of the genetic gold standard restriction factor level stuff. You can't, and you may or may not get that. That's one of the challenges of sort of starting with the evolution, is that you might identify really interesting relationships or robust connections, but it might be that the actual sort of infection biology or the virology that's happening isn't in humans. Maybe it's in fish, maybe it's in bats, maybe it's in another species.
Exactly right. Yeah.
And immediately the biological space gets so vast that it's hard, or maybe that pathway, the sort of let's say the equivalent of Elios, was really important 5 million years ago in our ancestors, but today less so. That like there are all these sort of
Sure.
Yeah.
I think, as you said, this is a good introduction to people. Say, here, look at these proteins. We think they may be important. Go study them. And now people need to do, not just in humans, but in other species, they need to do transcriptomics in infected cells and see if any of these have are going up in a way that would make sense, like you said, in your zebrafish. And not just one knockdown infect experiment. We need to do a lot more than that. But I think it provides a lead for people to do that.
Yeah, and I love this kind of from the standpoint of a lot of science, as we tell the stories, like the success stories, we often it's almost revisionist history where we give these really clean, like,
Yeah. Yeah.
here's the breakthrough discovery. Sea gas was that way. It was sort of this sought after sensor, and kind of changed the field, and is being now used in, people are attempting to translate this into real sort of therapeutic interventions. Most of science isn't that. It's this kind of stumbling around and saying, oh, well, maybe there's a link here. This is kind of interesting. And then you put
Hmm.
it on the record. And, I wouldn't say all the labs should stop the presses and study these three sort of sets of genes, but you kind of file it away and you say, oh, that's curious, or that's interesting. And then it might be 2 years later, you're doing a completely different set of experiments. One of GMAP6 comes to the surface, or, one of the other components in the alphabet soup here. And then you're like, oh, wow, that was tied to this and it sort of presents these links, this sort of like
Yeah. Yeah.
breadcrumbs almost. And then someday it's sort of like will take off. And this has that energy. I
Yep.
feel like, it's sort of, you see those links and you're like, oh, that's probably something, but we haven't quite put our thumb on it yet.
Yeah.
And
I
so,
mean, that's how science, we're not being critical. That's just the way
no,
science works, right?
exactly. And that's what I really like about this paper, is it really has that kind of like, well, maybe this, or I thought about that. And then but it has that kind of
Yeah.
stumbling quality in a positive way that could really lead to something down the road. And so, yeah, stay tuned in a sense in that way, or kind of have it on the radar as you're going forward. I would say a few take homes for me. Bacteria have complex immune defenses. I still can't get over this. Like in the last, almost, by the week, there's another paper that gets published about a new defense that someone sort of unearthed, these relics from bacteria. And in hindsight, it's probably not so surprising. There's so much diversity. There's so many viruses. You would expect that there would be all of this complex sort of, ways of contending or balancing out virus replication relative to host fitness. And so now with, I think, the breakthrough in the last few years has been, we have a ton of genomes of microbial genomic diversity available in public databases, and enough of a foothold to sort of use some of these patterns to then move really quickly into function. In a lot of ways, I'd say the field is in its infancy. We still know very little about the mechanisms. And there'll be a few, I would predict, that will really rise to the surface. There'll be sort of the next CRISPRs, the CRISPists, or whatever, that will be, maybe really somehow catch fire to kind of revolutionize molecular biology again, or to come into sort of translational space. But that's all we're kind of in the infancy of it. You can just see these things kind of bubbling up. And then, what happens next, I think, will be really fascinating. Yeah.
I want to thank Thomas for your contribution to science communication. Thomas says, I hope to donate more. I want to say how much TWiEVO and virology has
Mm-hmm.
changed my perception of viruses.
Wow. Thanks, Thomas.
And
Yeah.
here's a question we can ruminate on now. This is
Great.
good.
Let's do
Can
this. Yeah.
bacterial genes transfect and integrate into eukaryotic genomes?
Yeah. So yes, they can. Usually, there's probably an accomplice, a genetic accomplice at play, and that's where the viruses, I think, come
Mm,
into the conversation. So one of the places we do a little bit of this work, but not much. But one of the places where I think this is a fascinating sort of point towards is when we look at the genomes of giant viruses. And by giant viruses, we're not bluffing here. These are viruses that have genomes that are bigger in some cases than free living bacteria, have virus particles that are bigger in some cases than free living bacteria. And we start to just do a catalog of the genes encoded by these giant viruses. You find genes that you also find in the genomes of bacteria, archaea, eukaryotes. It's sort of like all of the above. And so one idea is that as these viruses have been replicating sort of or sort of professional gene collectors, that they're getting genes not only from sort of not just the hosts that they're in, but the other sort of the other species, kingdoms of life that are all kind of intermingled. And so for us, that might be, the close association with us bacteria or microbiomes. But for these giant viruses, a lot of them live in pond critters or ocean going eukaryotes, protists,
mm,
and amoeba and so forth, which are these professional grazer cells that are out there, feeding on bacteria and archaea, and then are infected with viruses or associated with viruses. And this sort of becomes the sort of cauldron for all of this gene exchange. Mechanisms, that's a great question, interesting question. I'm guessing a lot at play, and we're just sort of glimpsing at that at the moment as a field.
All right, Nels, you want to do some picks to wrap this up?
Yeah, let's do that. So my pick of the week, since we're on the sort of infection biology and in the personal virology, the sort of thread here my pick is pathoplexus.org. Put up the link on all our channels here. So Pathoplexus is a new open source database dedicated to the efficient sharing of human viral pathogen genomic data. And the hope is that this will foster global collaboration and public health response. So this is important, Vincent. So like, not only as we can take for granted that a lot of you just go to your, databases out there, go to GenBank or the major websites, NIH sort of repositories of genetic information that you can kind of get everything that's out there. And you can't in a lot of cases. And in fact, with SARS-2 in particular, we've seen some sort of bumps along the road. So for reasons that are still a little bit mysterious to me here, kind of is there's this database called GISAID, GISAID, which is originally put up to monitor, speaking of human viral pathogenic data, was doing a lot with influenza, possible pandemic influenza strains, and putting all of the genomic data up. But it's actually held by a private interest. And so for some reason, it was kind of in the right place at the right time as SARS-2, that pandemic emerged. And people, as they were sequencing the genomes or the genes of SARS-2, it sort of became the central repository. But because it's not, you have to actually, write to them and say, hey, can I get access to this data? And because it's not public, you're sort of at their mercy of if and when they'll allow you to have access to this data. So I think Pathoplexus is sort of an attempt not only to sort of democratize this or make this an open source scenario, but to also capture data sets from other viruses. And so if you go to that website now, you can see they have a few kind of links. So that, some viruses that are smoldering at the moment. So West Nile, there have been 7,000 new West Nile virus sequences added in the last month. Ebola in Zaire, there have been 3,500 sequences plus added in the last month. Crimean Congo hemorrhagic fever virus, say that 5 times fast, a couple thousand sequences added in the last month. So now, as these things continue to build, Mpox could be in there, other viruses, sequences. This then becomes hopefully a long-time curated site where it's a lot easier, sort of the barriers are lower to sharing this kind of data. And the value of that data, it's hard to tell. Like I think, collect data now and then sort of analyze later has been a pretty tried and true method for gaining a lot of meaning from some of these things, but it's really hard to know what's out there if you can't get access to it.
What do you think about that, Nels? If you publish a sequence, do you think people should have access to it, or can there be limits?
Yeah, no, great question. So data sharing here, in general, I favor the more open it is, the better. There are cases, for example, so let's say for people whose scientific enterprise depends on generating this data, and then there is definitely some priority kind of scenarios where having first access to that data you've collected allows you to say something about the meaning of that data, which then allows you to get more resources to keep generating that data. And so I'm totally sympathetic to that part of the situation. I don't think, there are cases where holding that data as you're doing sort of a first cut of analyzing it, but the shorter we can get, or, we can do that, or the more we can incentivize people to share first and then have the resources to collect more later, I think, to try to sort of take away that motivation to hide it or to have access to it first, that sort of the more sort of as a scientific community that we can make it easier or, less sort of scary to share data, I think the better.
Because a lot is not published, as you know, like many viral genomes are just sequenced as part of surveillance and they're put in a database. So it's not clear, how, but, it seems to me that the people should not control these databases, right? You should say this is an open database, which I think is what Pathoplexus is meant to be. If you put
Correct.
it there, it's going to be shared. So if you don't want it
Yes.
shared, don't put it here, right?
That's right. And so then as someone who's, weighing, should I put my stuff there and how fast? That's where
Yeah,
if we can lower that barrier by making to incentivize sharing, and because that unlocks, so a lot of it will be necessarily sort of worthless, right? It's kind of needle in the haystack sort of situations.
of course.
But having it, not even like needles in a stack of needles. I mean, it'll, or whatever. But the you know the ability to analyze it increasingly with more sophisticated machine learning tools, et cetera.
Yeah,
If that
yeah,
if that brings something to the surface, a concept that might not otherwise be possible. I think it's like the more that we're doing this, the better. But but I get it. It's complicated.
the best example is the first SARS-CoV-2 genome sequence done in China,
Yeah.
right? Eddie Holmes convinced them
Yeah.
to share it because immediately people started using that to make vaccines, and
Yeah.
the result is amazing, right? So I mean, not everything is going to be like that, but you
No.
just don't know. And if something is, it's a sequence, it's a biological organism. It's not something you made, right? It's not
Yeah.
some proprietary thing that you want, an enzyme that you've modified. Just put it out there because humanity will figure out what to do with it.
I agree. Yeah. Exactly right. Yeah. Yeah. Yep. So anyway, that's my pick of the week. Vincent, what's your science pick of the week?
So this is an article in Nature. It's called "Can Science Cure Its Addiction to Plastic?"
Hmm.
This is something that's bothered me for my entire career. So at the moment, labs use amazing amounts of plastic and they
Yeah.
throw it away. Most of it gets burned. I mean,
Hmm. Mhm.
pipettes, vials of all sorts of sizes, pipette tips, cuvettes. Also, I mean, I can't
I
even
know.
begin to name. And I used to, for example, we have these 50 ml tubes, the centrifuge tubes.
Yeah. Yeah.
Hundreds a day just get thrown away.
Yeah.
And it always bugged me because when I started in science, everything was glass. There was no petroleum byproduct industry, right?
Yeah. 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 Yep.
We had to wash pipettes and glassware and vials and reuse them. And when I started my lab, we had lots of glass pipettes and glass bottles and so forth.
Oh
And
yeah.
at one point, the university said it's too expensive to wash these because we used to send it upstairs to a
Mhm.
washing place. They would wash it and autoclave it. They said it's cheaper to buy plastic and throw it away.
Yeah.
Okay, I understand the economics, but man, what what a huge consumption. So this
Yep.
article is all about some efforts in the UK to try and get on top of all this usage. Um, this is an article in Nature in 2015 estimated
Mm.
that 5.5 million tons of plastic waste was being generated yearly by labs worldwide. That's
Mm.
2015. I'm sure
Yeah.
it's
More.
much higher now. And so as long as I had a lab, this really, really bugged me. I would see people just taking plastic and,
Yep.
left, right, just throwing it out. And I don't think it's right. I think we need to do something about it. And, sometimes it's not just about economics. Um, and yeah, so Tryon says
Yeah.
glasses break easily. I understand that for many things, but pipettes, Nels, you probably never used a glass pipette, right?
No, no, I'm not that young. And in fact, we use glass for some of our stuff. We use glass pipettes now, and we have a dishwash on our floor
Hmm.
with for all of our. But you're right. I mean, we also go through a ton of plastic. This is increasingly, really getting under the skin of folks in my lab, myself included, and we're kind of so far down the road with plastic and a lot of stuff. But can we, and then recycling plastic is a whole nother, third rail here of economics/environmental stewardship
Yeah,
as
yeah.
well. Thinking about the news headlines just yesterday, the state of California suing Exxon for the some of their false ideas about how recyclable plastic is or what's been happening in those pipelines. But so anyway, folks in my lab, we've moved towards all our pipette tip boxes. We don't throw them out; we save them, and then the pipettes tips get reloaded
Mm-hmm.
into them. It's sort of a small. We have some recycling, presumably recycling. They seem to be under reputable sort of thing. So we're trying to do a little bit better. We can't do it all. If you're using a plastic pipette, for example, in something that's a biohazard, then
Mm-hmm.
recycling, if it's BSL-2, we've been using a virus or whatever, something like a
Sure.
microbial agent that sort of changes the equation. Then we get into this awful scenario, as you're pointing out, of burning, or being, autoclaved, et cetera. And so anyway, yeah, this is a tough one. And your point, I think, is a really important one, Vincent, about, when we think about economics, what's cheaper for a university today, but what's more expensive for our communities in the long term, as we're starting to bring into focus the impact of microplastics, both how pervasive they are in the environment and how that is going to impact our own health with some pretty substantial economic implications. And so, yeah, the more we can do to maybe not cure the addiction, but at least manage the addiction to plastics, I
I
think
want
that's really
people to be
important.
aware of it because I fear
Yeah.
as new personnel come into labs, they don't think twice. They say, "Oh, everybody's using plastic and just throwing it away, so I'm just going to do the same thing." You have to
Yeah.
think about what you're doing, right? You're contributing
Great.
to an enormous issue, and I just want you to be aware of it, if anything. So
Yeah.
this is a good article that discusses it, and it's always bugged me. It just has always bugged me.
Yeah. Plastics aren't going away. And this is the kind of one of the major
Yeah,
pillars of technological challenges that we've presented ourselves, whether it's, the ozone hole 50 years ago, 20 years ago, which seems to be turning in a good direction. Climate change, which is another massive one, our use of plastics and what that
yeah.
means for environmental and health impacts. Yeah, some pretty major challenges sitting on our
I
plates.
even had tissue culture glass pipettes because they're shorter, right?
Yeah.
Because a 10
Oh, yeah.
ml pipette is pretty long, and in a tissue culture hood, that's too unwieldy. So they made short, fat ones that you
Yeah.
could use in the hood. I love them. And they
Yeah.
said, nope, we're not going to wash them for you anymore. It's
Um,
like, this is the thing. When you're at a university, you kind of have to do what they tell you because they're supplying the building and
yeah.
everything,
The
right?
infrastructure. It'll be
And
interesting to see if some of those calculations change going forward, right? That actually it's, if there are penalties about how much plastic you put into the environment,
well,
what does that mean, right?
as you said, it's about the future cost. I
Mm.
mean, as far as I understand, plastics are a byproduct of petroleum, right? So it would be thrown away. So it's almost cost
Yeah,
free, right? Not cost
that's
free,
right.
but it's cheap. And so that's the
Cheap.
driving force. And I agree for contaminated stuff, it's great to be able to throw something away and burn it. But,
Mm.
you know, not for everything. I mean, pipettes in
No.
particular, right at the bench, you take 5 ml of sodium chloride solution with a plastic pipette and you throw the plastic pipette away. That's ridiculous.
Yeah, agreed. Yeah, yeah. Yep.
Hey, by the way, we have one question from Thomas, which we
Oh,
should
great.
answer here.
Oh,
Would it
good.
be safe to publish gain of function research like this to the public? And so there is a little aspect of gain of function here, and it's published. What do you think, Nels?
Oh, for the paper that we covered today?
Yeah, the paper that we covered. Yeah, they mentioned
Oh,
gain of function actually
oh,
in
do
it.
they? Oh, interesting.
Yeah, we'll
Yeah.
find it.
So for me, that didn't immediately, great question, Thomas. Didn't immediately jump to mind. So, for example, when I'm thinking about gain of function, um, for example, for like messing around with the genetics of infectious microbes or viruses, and then sort of seeing how that impacts. They're working on the other side of the coin here, which is, so they did make, like, the closest I would say to gain of function is that chimeric protein we described. So they took part of
Right.
the immune defense gene, the ileos system from bacteria, and then spliced that together with the rest of the GMAP6 gene that we have. So the gain of function here would be, it would make potentially a better immune gene, which would actually, in terms of safety, the more sort of supercharged immune genes we might engineer or come up with, that would, counteract the, current infectious microbes, viruses that are out there. And so, this is the kind of gain of function work that, and it's important actually to sort of, as we define gain of function, to remind ourselves how positive gain of function can be, um, if it's especially if it's practiced towards enhancing immune functions. If in this case you might say, oh well, maybe they'll make a crappier immune gene, and that's probably true actually, because they're sort of splicing stuff together here. But they're not changing anything about our own immune systems. We're not changing our own genomes here. This is all done in cells, and it's it's not going into humans or into germlines or even into animals. And so there's really, I don't see any risk here of gain of function. I guess if you really squinted at it and wanted to try to get something, you could say, well, if you design a new immune gene and then the viruses sort of figure out how to go against that, and that, indirectly there's sort of this knock-on effect. That's really grasping for a sort of a rare probability, sort of stacked on rare probability, stacked on rare probability. And so, yeah, for me at least, this is very safe to publish. The advantages far outweigh the sort of minuscule possibility of something bad happening. And again, I think getting this kind of information out to sort of the breadcrumbs of thinking about how do we understand the world around us, the immune systems around us, the more we're sort of adding to positive scientific knowledge.
Remember, most gain of function research is absolutely fine. There's just a tiny fraction that raises some eyebrows, but people have used that to define the whole word as being unsafe, and it's not correct. Nels has been doing gain of function his whole career, and most of it's just fine.
That's all. Well, genetics is that's more than half of genetics, I would say.
Yeah,
gain of function. So we need to try to kind of reclaim that term, because it's sort of gotten. I mean, this is anyway.
Yeah, it's a good. Yeah, we have to take back gain of function.
Yeah, gain of function is good. We want to gain functions. Gaining the ability to speak in our this human language. What a great gain of function for our species. Bipedalism, the ability to walk and get around. What a great gain of function relative to sort of some of our ancestors.
Flight.
That was a pretty good gain of function breakthrough for bats and birds and other critters, insects, etc. And so, gain of function is, by and large, is like sort of the fuel of evolution, the sort of progress of biological life.
Okay, that will do it for TWiEVO 106. You can find show notes at microbe.tv/TWiEVO. If you enjoy these programs, we'd like to have your support. You can go to microbe.tv/contribute. And Microbe TV is a nonprofit entity in the U.S. That means your contributions are U.S. federal tax deductible. Nels Eldy's at cellvolution.org. Early Bird on X. Thanks, Nels.
Thank you. Loving the live stream. Thanks for all the questions, everyone. Thanks for coming along with us on another science adventure.
I'm Vincent Racaniello. You can find me at Microbe TV. I want to thank our mods for stopping by today. Andrew, Barb Mack, Les was here, Tom was here, and who else? Anybody else? I think I named everyone. We appreciate your comment. Tonight we have another live stream, 8 p.m. Eastern office hours, if you want to join us here on the same channel. And the music, by the way, on TWiEVO is by Trampled by Turtles. You can find their work at trampledbyturtles.com. We'll be back next month here for TWiEVO 107. Check out the Microbe TV website for the schedule. Till then, everybody, thanks for coming, and be curious. Bye-bye.